altogether. Published the paper in Nature.
Cell biology.
Cell biology. Yeah, always mix up the communications. But what Mia is presenting to us is actually now a follow-up on this paper or on some of this work, because it was her work, she was the first author on that paper, that got us involved in a better understanding of what aneuploidy really means in implantation stage embryo. And so it's my real pleasure today to welcome Mia back home. And she is going to talk about Prone Human Embryogenesis. It's a latent power chromosomal abnormality. And Sam, Ali, and I, we have been very interested in micronuclei, and some little bird told me that we will hear also a little bit about micronuclei. Yeah, great having you.
Thank you so much, Robert, for inviting me back to New York. I'm really looking forward to this one today. And you're right, it's always like a homecoming to be here. And also, I'm so glad to see so many familiar faces and my mentors and my colleagues. So I'm really glad you're here to attend my talk. Anyway, so today I'm gonna talk about some previous and new findings about this research, our major work in aneuploidy. And today my talk will be the Prone Human Embryogenesis. And I will share with you some findings of our research in the sulfate and tolerance of the chromosomal abnormality or aneuploidy. Anyway, so I believe some of you are clinicians or medical doctors, so I don't have to stress how inefficient human reproduction is, as 1 out of 6 people globally facing the infertility. And in my 30s right now, I think I'm young still, and I'm defined as young. You don't need to applaud for this, but anyway, so anyway, I'm defined as young in modern society in every sort of way. I'm a young scientist, junior PI, anything. But by my OBGYN doctor, they claim me as the advanced of maternal age, and I literally offended by this. And that tells you how inefficient human reproduction is. At young by age, you already kind of get to the point, like reproductively, you're slowing down. Anyway, so today I'm going to talk to you one of the root causes of the less inefficient human reproduction, which is on the embryo level. So basically, one root cause is the nature of the prone nature of human development. And basically, by now the detection method for pregnancy is more sensitive. So now we come to a realization there are more miscarriages than we previously know. So basically, you see this whole human developmental process by just stage 23, and you can see like by the first 2 weeks of the fertilization, almost half of the pregnancy will be lost. So the women will not even know they are miscarrying when because they don't even know a lot of them don't even know when they're pregnant. So only 1/3 of the pregnancies can pass the first trimester. So, in China, there's always a saying from the older generation, you don't tell anyone you're pregnant until 3 months later. I think they actually have a reason, because most of the pregnancies will be lost. Anyway, so among all the reasons, the chromosomal abnormality is undoubtedly one of the major causes for the high mortality of the embryo. And basically, the cells that carry the chromosomal abnormality, we call them aneuploidy. And aneuploidy happens when chromosome missegregated during either mitosis or meiotic stage. So when it happens to the meiosis stage, basically in the gametes, either sperm or eggs, the downstream embryos will basically have all the cells that carries the abnormal phenotype. So in this case, majority of them, almost mass majority of them, is not going to be viable, so they are lethal. The only exception will be some disease like Down syndrome, Edwards syndrome, tumor syndrome. Okay, so but when it happens in the meiosis stage, actually most of the missegregation of chromosomes happens at the early stage of embryo or after differentiation. So when it happens at the mitosis stage, as you can see, only partial of the embryonic cells will be affected. So this will lead to the embryos carries both normal cells and abnormal cells, and we call this phenomenon mosaicism. And surprisingly, with the more advanced detection technologies with the chromosome status, we come to a realization that chromosome instability is actually very common in human cleavage state embryos. And from some IVF studies, they show that more than 70% of the embryos are actually mosaic embryos that carries normal and abnormal cells. So, and surprisingly, a lot of those embryos can actually lead to the live birth. And I'm sure Norbert can tell you all about this, like because the experience of transferring those embryos to the patient. However, in the natural pregnancy, this is also the phenomenon as well. So basically, it means the embryos have some kind of fitness selection going on that actively eliminates the abnormal cells during the development. And as a scientist, as a basic scientist, my curiosity arises from why this happens and what happens to the abnormal cells during the development. So hopefully, the study reviewed from those basic research can give the clinicians or medical doctors some basics of what happened to those normal cells, so make better adjustments of how we handle those type of embryos. Anyway, so in today's talk, I'm gonna basically show you some previous and current findings of our research, and I'm gonna break it down into 3 sections, which involves the aneuploidy in pre-inclination embryos, aneuploidy in the post-inclination differentiation, basically after the embryo implants, what will happen to those organisms. And lastly, I'm also gonna show you the aneuploidy in the extraembryonic tissue, which is the placenta, and how the aneuploidy are doing stuff there. Anyway, so basically from today's talk, since we are a stem cell based laboratory, at Upstream in our Leeds lab, basically a very state of art stem cell hardcore laboratory. So I pass on this legacy of continuing my stem cell work in that laboratory as well. And so based on this, we decided to tackle the aneuploidy sulfate question using the stem cell models and also stem cell AI. So today you will hear from the talk mainly 3 embryo models. So one is the blast weight model, which generated from the stem cell that kind of recapitulates the early pre-implantation stage of development. And we also will have the gastrulation model, which models the post-implantation perspective of differentiation, which involves some gastrulation events like the 3-joint layer formation. And lastly, you will hear about some findings in the trophoblast organoid, which basically recapitulates some early perspective of the placenta development. So first, let's talk about the aneuploidy in the pre-implantation embryos. So in a couple years ago, so this is like Norbert say, it's a collaborative project of Ali's lab and CHR. So basically, we decided to look at the chromosome status in the embryo using some single cell level data. So we think that the aneuploidy prevalence is more than what we thought before because the detection technology is not the solution where it can detect the single cell level aneuploidy. So in order to do that, to make a more precise prediction, so we look into the single cell RNC data back then was available in some in vitro growth human embryos. So, so since it's a single cell RNA-seq data, it's not going to be as accurate as the single cell whole genome sequencing data. However, this method was developed and tested, so the recording efficiency is actually good enough for us to make rather accurate prediction. So anyway, so using this data, and the principle of the strategy is basically the dosage effect of the transcript. It's basically if you have more chromosome, you will have more RNA transcript coming off on this chromosome. So the RNA data will reflect to some level the the dosage of the chromosome. Anyway, so using this method, we have found that the early stage embryos at day 3, they are actually very, very high. Right? As you can see here, it's almost 80% of the embryonic cells are aneuploidy. And you can see they drastically decrease over time. And by the time of the blastocyst stage, it's already dropped significantly. So this basically means aneuploidy happens most frequently in this earlier stage. And recently, I also came across a clinical paper where they show that when they do the biopsy on the A cell stage in the addition of aneuploidy, then when they do the biopsy, do the testing on the blastocyst. So that's a paper I come across recently, and I think this also confirms our hypothesis that aneuploidy is more frequent in the early stage. Anyway, so the next question. So we're gonna tackle is we want to kind of see why this is the phenomenon, and then we decided to use the stem cell as a model to tackle this. So basically, if you are not a stem cell biologist, you might not know that there are different types of chlorophyll stem cells. So the regular or conventional chlorophyll cell most people refer to, they are actually this type. So it looks flat, nice quality. So this is the prime stage embryonic stem cells. So basically, they resemble in human the prime stage chlorophyll stem cell. They resemble the post-implantation epiblasts, basically the after implantation, the embryonic cells. But we want to kind of study the early stage. So there's protocol available to convert this type of cell to the early stage, which we call them, the other figure there, we call them the naive chlorophyll stem cells. And this basically, these cells are higher up in the pluripotent hierarchy, and they have higher ability to develop into any type of cell. So if you have the chlorophyll stem cell, they are able to contribute to any cells in your body. But for the naive cells, they can even contribute to the placental lineage. So they have higher chlorophyll and basically the early stage. So when we convert the prime ES cells to the naive cells, we realize along with the potency level increase, they actually have much more chromosome instability going on. And those cells are just chromosomally unstable. During the culture, they just accumulate, constantly accumulates those chromosomal abnormalities. And basically, by the passage 20, almost all those cells become aneuploidy. And also, the tolerance level of aneuploidy in those early cells are actually much more than the prime stage embryo. Because when we try to convert that, the naive cell line that carries aneuploidy cells, when we convert them back to the prime stage, all the aneuploidy cells actually get eliminated along the way. So this basically means that those cells are inherently unstable at early stage. And also this is it's a conserved phenotype for primates as well, because we also look at the monocyte embryonic stem cell early stage and later and same patterns of this phenotype. And my technician, my research scientist in the lab, Andrew, so he decided to look into the transcriptional patterns of those cells to see if there's some insight we can review from those early stage cells. So one thing really interesting we found out is that we look at the down-regulated pathways of the naive chlorophyll cells, and we found the mass majority of pathways that got affected were actually the cells that involved in the cell chromosome segregation process, the cell cycle process. For example, you can see the spindle assembly checkpoint genes were down-regulated. So we believe this could be the intrinsic reason why such cells are highly unstable. And then another interesting we found is that when we look at the upregulated genes in those early cells, we found among all the total genes we expected to see, we also saw a lot of upregulated pathways that related to the process of autophagy. And as you might know, autophagy is the process of the cells to actively recycle or basically degrade the toxic stuff in the cell to help cells either survive or apoptosis. So we found this highly upregulated in the early cells will they have unstable status, which means this could be the reason why such abnormal cells are tolerated. So, anyways, but this is also interesting because a recent study in Jennifer Gold's lab from, I believe now in, anyway, previously in Cambridge. So her lab, they found that in mouse, autophagy actually induced the apoptosis of the embryo. So autophagy is actively kills the abnormal cells. But this, we found the other way, and we think the autophagy is help the abnormal cells to be tolerated. So this discrepancy in data in mouse and human could be some evolutionary difference among species, or maybe it's just autophagy has dual meaning, because in cancer, autophagy is a double sword word, double-edged sword. Basically, it can either help cancer to survive or humans. So maybe they're just more complicated process we need to further dig into. Anyway, so then we decided to use this naive stem cell to look at some dynamic change of aneuploidy in the world. And this is a model we use to study this. So in recent years, there are more advancement in human embryo model. And a couple of years ago, so Nicholas Ribon's lab, they developed a protocol to use the naive stem cell to self-organize into the embryo-like structure. So this is the methodology we adapt, and we use basically see the naive pluripotent cells in those micro walls. They're pretty much just the V-shaped walls, and the cells will self-aggregate together. And with some like signaling cues, the cells will self-organize into the structure with just like the field. And they if you look at the staining data, basically you can tell that the middle cells expressing four is the pluripotent part as immunosomas, and the outer layer will be the campus positive trabectolate. And those embryos are resemble the real human embryo also by their attachment to the dish, and they can further grow, and the outer layer will chromoblast or trabectoderm will further differentiate and kind of become formally functional as you can see as the human chromatography, as you can see in the video in the doctor's video, anyway, so, but here I want to stress that those human embryos are not anything close to a real human embryo. So they are basically can model some aspects of the human embryonic development, but it's nothing close to being functional. So as a scientist, we use those models to understand some perspective of the biology, which come in handy for this model system. Anyway, so we found that if we're using the aneuploid naive cells to form the blastula, nothing affects the formation. So we didn't see any difference. And so aneuploid can pretty much form the same structure as the normal cells. So then my scientist, Chong, so she decided to look at what happened to mosaic when there are mosaic situation going on, or what happens to those mosaic embryo models. So she decided to see together the aneuploid cells with normal cells. As you can see, the abnormal cells here are the J cells, and the normal cells are the blue ones. So we can still form the blastoid, but you can see the majority of the inner cell mass, which is the embryonic period, shows the green normal cells instead of the abnormal cells. However, this is only the preliminary data and need more quantification. So for the future, we will study more precisely, like the allocation of aneuploid in this condition. And also, we were gonna do different ratio of aneuploid mixing together with normal cells in this model to see if there's a threshold of tolerance to this aneuploid. Anyway, so on this part of research, we can confirm that the pre-incarnation embryonic cells are constantly unstable, and aneuploid in those early stages can be well tolerated, and it seems like there's a dynamic of tolerance change when they get differentiated. And it seems like the existence of aneuploid doesn't significantly impact the lineage specification. So now let's move to the part 2, the aneuploid in post-incarnation differentiation. So this part of work was done in Ali's lab, collaborated with Norbert, so that was my support. Anyway, so for this work, we use a model system, elegant model system developed years ago in Ali's lab. So this is called 2D micropattern gastrulation. This is a very elegant system because the cells can be confined to the geometry, confined to the chip and creates some confinement with geometry. And with some certain signaling cue, in this case the BMP4, the cells can be self-organized into this very nicely organized micro gastrulate system where you have the cells in the middle express the SOX2 positive endoderm marker, and you will have the BRCA positive mesoderm marker on the in the middle, and then lastly you will also get some extra embryonic cells in between the model expressing the so as I think this model basically is geometry confined, so it's very easy for computation data. So we saw this as a great model to dissect the surface of any code, a lot of differentiation. Anyway, so basically in time state, so I forgot to mention this model was derived from the time state embryonic stem cell. So the Glasgow model was derived from the naive state. So time state chlorophyll stem cells, they are actually pretty stable in their concept. So in order to create the aneuploid chlorophyll stem cell, not like naive cells, they just naturally accumulate it. So in this case, we have to treat them with drug to force them to become aneuploid. In this case, we use this MPS inhibitor, basically called reducing, to kind of activate the single assembly checkpoint to force them to become aneuploid. So we can confirm after the treatment, most of the cells will become aneuploid. As you can see, the normal cells there, they show the 46 chromosome number. However, the resuscitated one, even after 24 hours, you see a lot of like chromosome presentation going on, weird crossover there, and we confirm by doing some flow cytometry to see the DNA contact is also significantly changed. And by the time of 24 hours treatment, almost 100, 94% of the cells will become. So we wanted to know like what happened to those aneuploid cells when we are trying to form this embryo, or not embryo, but more like when they are doing the differentiation of the embryonic cells. So as you can see, the normal cells, they are forming the gastrulate very nicely with the beautiful pattern of the extra embryonic layer in the middle part of the embryonic filament with the ectoderm, mesoderm, and endoderm. But when we see the aneuploid cells on this pattern, what you see is that while forming this gastrulate, you can see the middle part of the embryonic domain is totally ablated, and then that's the GATA3 positive green extra embryonic cells. They remain intact. So we also confirm this is not a density-related issue by basically double seeding more cells than needed, because we need to make sure the cells are complementary of each other, creating this kind of interaction going on. But even with very dense population, the cells failed to form the nice pattern of the gastrulate pattern. So we know that aneuploid cells are preferentially differentiated into the subacromial. And next, we want to know what happens if the cells already took a certain fate, if they're aneuploid, what will happen to them? So in order to study that, we basically treat the micropattern embryonic stem cell with BMP4 in the same time as reducing. So the rationale is that the BMP4 will differentiate the cells instantly. However, reducing will take the whole cell cycle for them to become aneuploid. So in this way, you can know how aneuploid behave after they take a certain managed phase. So in this case, you can see while forming the gastrulate, the aneuploid gastrulate, there are still 3 layers forming. However, there's significant decrease in the middle part of the gastrulate, meaning the embryonic phase is just more affected than the outer layer of the embryonic extra embryonic phase. And you can see there are a lot of cell degrees going on in the middle, indicates some activity of apoptosis. So we also confirm that with the apoptosis marker p53 and cell death marker cas3. As you can see, p53 is more a cell damage marker or stress marker. They are everywhere. However, the cas3 cell death marker is highly concentrated in the middle part where the embryonic domain is. So basically, the embryonic domain is more effective, more actively deleted when they become aneuploidy. And aneuploidy in the outer layer, even though they are also stressed by the red marker, however, they are well tolerated. Anyway, so, and then we decided to see what's going to happen if they're in the mosaicism situation. So basically, to do that, we mix, same as the mosaic gastrin last week, we mix two population of cells together, and color those fluorescent tabs so we can differentiate them from each other. Basically, if we if we have 100% control cells, like the normal cells, which are the green cells here. Oh, by the way, each dots are a cell. So if we have like the control cells, all normal, the gastrulate rate look normal. They form this very nicely pattern, and these cells are aneuploid cells. If all cells are aneuploid and pink there, and you can see the pattern was totally distorted. However, if we add in 25% of normal cells into the aneuploid gastrulate, you can see the pattern was significantly rescued with the middle part of the embryonic phase restored. And if we increase the number to 50 or 70%, you can see the method pattern look more like the normal type pattern. So the phenotype of the germ layer formation was rescued by adding the normal cells in this. And then if you look at the live image of the this differentiation process, you can see that basically green cells are normal cells and then we're seeing treated aneuploid cells are pink cells. As you can see, during this 48 hours of treatment, during the differentiation, the green normal cells totally outcompete the abnormal pink cells. So we believe this process of rescuing is likely by cell conjugation, by actually eliminating the abnormal cells. And with normal cells dominating the pattern can get rescued. Anyway, so we also found the apoptosis of aneuploid cells is dependent on the differentiation process, likely with the BMP4 induction. As you can see, without BMP4, the cell number, so those are all aneuploidy seated on the right side. So without BMP4, you can see the red marker is the apoptosis marker, and yellow markers here, they are the cell death. So those are cell death. You can see without BMP4 that there are much less dead cells with those aneuploid cells. But with BMP4, you can see more cell death in those combined area. Anyway, so if you look at the quantification, the cell number also significantly decreased in the ones that aneuploid cells were BMP4. Anyway, so remember I was talking about the autophagy we observed in the non-stable naive cells, so we want to see if actually autophagy plays a role in the aneuploid colonies. And micropattern is such an elegant model, so we decided to try first in the micropattern gastrulate. So basically, while we are inducing the, so this is an aneuploid gastrulate. They look good, right, with the normal embryonic parts really small, and this is with gastrulate forming with this drop called rapamycin. So this drop basically blocks the mTOR pathway and induces autophagy to happen. And with this drop alone, it doesn't seem to affect the gastrulate formation. And if we apply the rimacin together with rapamycin, basically we try to upregulate the autophagy in the aneuploid gastrulate formation, we can see that inducing autophagy actually rescue the phenotype significantly. As you can see, the middle layers was significantly restored, and they're much less cell death if you look at the Rabi staining of the DNA. Anyway, so this is just very preliminary results. So we basically think that autophagy is contributing to the cell survival of the individual. So basically, this is the second part of our work in the antibody in the post-inflammation differentiation. And we found that antibody cells are basically dynamically eliminated during this early post-inflammation differentiation along with gastrulation and other differentiation. And this also from this micropattern data, we also reviewed that there's some preferential tolerance towards the aneuploidy in the extra embryonic image. So our next question is just how aneuploidy are doing. How is aneuploidy doing in the extra embryonic tissue or placenta tissue? And placenta is such a unique organ because it's temporary. A lot of people just ignore placenta, but it's actually really important for the success of the pregnancy. It's essential for the success of the pregnancy. And you will be surprised how understudied this area, placenta, is. There are not many advanced study actually currently available for placenta. So I think this is definitely a field that worth exploring more. And we definitely hope to contribute to this field more. And anyway, so placenta is also interesting fun fact. It's the least evolutionarily conserved organ. So if you look at different species, mouse and human, they are very different in their placenta. And this human has a very invasive placenta that deeply rooted in the uterus of the woman. Anyway, so it's a very interesting organ, and that's understudied. And a recent paper, not recent anymore, 5 years ago, so there's a Nature paper coming out. Basically, they did some bulk sequencing on those placenta. Basically, they found a very extensive level of somatic mutation in this organ. And that almost equivalent, like the level of the mutation is almost equivalent to the childhood cancer. So this pretty much mapped the placenta a cancer-like organ. And if you think of placenta, they're invasive, they modulate the immune environment. To me, I feel like they're just a temporary cancer going on, you know, but they are not. They are tightly controlled. They are doing their function very nicely. So we want to kind of see, since we see this high elevated somatic mutation, we want to also see the, chromosome-wise, chromosome level, if there are any abnormalities. And also from this paper, because it's both level sequencing, so they cannot tell like if the cells are paratypically, chromosomally abnormal. But they, from their data, they suggest that means the any could be very often happening in placenta. But that's something we want to look at. That's why we decided to look at the placenta on their cell level to see if there are any. Anyway, so then we decided to look at the human placenta data recently published last year, where they used a single nucleus metastasis, which is basically a sequencing method that detects the open chromosome area of the cells, of the DNA, of the chromosome. Anyway, so we adapt methods which can predict the copy number values on those metastatic data and predict the chromosome type of the cells. And since it's single level data, so we can kind of predict the aneuploidy prevalence in those cells. And the methodology is basically saying the copy number values. And you can see this is an example of aneuploid cells in placenta that shows the present 18 chromosome 19. Anyway, so we look at the basically this time I want to also stress, I forgot to say, so their data so are the decidual or placental sample from the normal pregnancy. So those are all normal pregnancy aborted sample, no miscarriage or other pregnancy complication. So by our previous understanding, we just thought normal pregnancies placenta should have low aneuploidy. That was our assumption, but moving forward, we saw actually, if you see this paragram, all of this techniques, you can see this is the blood sample. So we also, as a control, we profile the blood sample. So anyway, so each line of those, they are actually raw cells. Green shows there's no change. They're also just chromosome for each chromosome. Anyway, so there's most majority of the cells are green, which means they're normal cells. So this is the green. if you look at the placenta sample there, you see those red lines. So on different console, each one of the columns and points. So if you look at the placenta, the 8 weeks placenta, you can see a lot of red lines indicates the additional chromosome. So anyway, so we see a lot of cells in the placenta have chromosome abnormal. And basically we looked at it and we saw almost 10% cells are placenta in the every cell type in the chromosome. And also I want to stress the accuracy, the sensitivity of the ataxic aneuploidy detection is not as sensitive as a human genome sequencing data. So based on the paper methodology paper, they say the precision is very, very high, 90-something percent, but sensitivity is low. So this means that we saw 10%, but it's definitely going to be much more than 10%. And it's very interesting. So the paper was under revision, and one of the very knowledgeable reviewer actually pointed out maybe we should look at the subtype of placenta, because from some previous paper they showed that when placenta or trophoblast cells get into a more invasive phenotype, they will require more chromosomal upload. So we took the advice and looked at the data in the subtypes of the trophoblast, and we found that basically I'm gonna show you. So the trophoblast, there's a cyto trophoblast, extravillous trophoblast, and syncytial trophoblast. So the cyto trophoblasts are basically trophoblast stem cells that produce all the subtypes of trophoblast. And the EVT, the extravillous trophoblasts, are function as the invasive part, help the embryo to dig into the uterus. And the syncytial trophoblast is for hormone functions, they produce hormones. So the assumption is this one will have more aneuploidy. And look at that, we definitely have much more aneuploidy detected in the EVT than the syncytial trophoblast. And syncytial trophoblast has the lowest aneuploidy level, which also makes sense because they need the transcription to be like very accurate to do their function of producing hormone, while the invasive trophoblasts need to be invasive and reproduce. That's why they are more aneuploidy along the way. Anyway, so from this data, we are just really think the placenta is an inherent feeder of the aneuploidy is an inherent feeder of placenta. And also, if we look at by chromosome, you will see that I found this very also interesting is that the most frequent abnormality happens in those chromosomes that can be variable in the aneuploidy cases. If you look at trisomy 18 Edwards syndrome, 21 Down syndrome, those cells, those aneuploid cells, they are fitter, things like even in placenta than the other type of aneuploid. Anyways, and also in this placenta, it's just different type of aneuploidy instead of just one clonal type. That means this is not just one single event. This means the chromosome will absolutely like having abnormality. We call it chromosome instability. Anyway, so my postdoc, she decided to tackle this question also by the stem cell model to see what causes defects, like why the placental trophoblast cells are so unstable. So we decided to use the trophoblast stem cell. So in recent years, people have developed protocols to kind of derive from the naive stem cell to differentiate into the trophoblast stem cell, equivalent to the cytotrophoblasts in the placenta. So we decided to use this model, these cells, to tackle how aneuploidy is doing in the placenta. And those cells has the differentiation capacity, as you can see in the organoid model. They can differentiate into the pink trophoblast, EVT, which is the extravillous trophoblast, and that's invasive. And they can also differentiate into marked by blue, the syncytial trophoblast, which produce hormone. So interestingly, we detect a very high level of DNA damage in the trophoblast cells. And as you can see, marked by this marker called gamma H2A, it basically stings the double-stranded break in the cells. As you can see, a lot of the trophoblast, on high level of the cells actually have a lot of DNA damage going on. And you can ignore the different type of trophoblast stem cell listed here because we want to make sure this is not an artifact by the in vitro induction. So we use all different type of the induction method to make sure this is the lineage specific event. Anyway, so we observed a high level of DNA damage, and then we decided to look at the chromosome level. So another technician in the lab, Katie, so she decided to accurately measure this by FISH, the fluorescence in pseudohybridization to detect the chromosome. And we tested 3 chromosomes, I believe. Here's only 2 for data. But anyway, so we see that the abnormalities in the early passage trophoblast stem cells is high already. But then along with the passage number, the aneuploidy actively accumulated, and you got even higher number of chromosome abnormality going on in those trophoblast stem cells. Also indicates the chromosome is highly unstable in the trophoblast cells. We also try to look at the heterogeneity level. When we're doing the whole genome isolate sequencing, which can be used to predict aneuploidy, on the bulk sample of the trophoblast stem cell, we didn't see any difference because the heterogeneity of the cell, they have all sorts of aneuploidy types, so it's almost impossible to detect the chromosome status on the bulk sample. So we decided to move with the single cell RNA combined with the single nucleus the taxi to give us more insight of the heterogeneity of the population. So what we saw is, there are actually 20 to 30% of the cells are aneuploidy with all sorts of different aneuploidy types. And this also indicates the trophoblast are highly unstable chromosome. And then, inspired by the work of Sam on the micronuclei, we decided, because micronuclei is when the cell segregates, if they're aneuploidy, they likely will have some lacking chromosomes and some fragments of the DNA, and those things will kind of envelop in the small nucleus. As you can see here, the blue area is just the whole nucleus, but this little dots here, they are like the micronuclei with some DNA contents of stuff wrapping into a small tiny dots here. Anyway, so this basically shows, this is an indicator that the chromosomes are not stable in the cells. And we see that in all the trophoblast cells we derive, the micronuclear percentage is very high compared to the very stable in the prime stage pluripotent stem cells. And this is also an indicator of the cells are highly unstable. And then very surprisingly, we found even with such abnormalities going on in the trophoblast cells, those cells are perfectly fine. They proliferate really well. As a matter of fact, some very severe aneuploid trophoblast clonal line, they actually proliferate even better than the normal one. If you're interested, I can show you the data later, but I'm not going to go into that. But anyway, so those cells are proliferating well, acting normal, and they can also differentiate into the upper one, the yellow cells, the EBT, and lower one, the hormone-producing ST. So those cells are functioning normal and has no effects by the aneuploid. So this confirms to us again, like aneuploidy might be just a normal figure. It doesn't affect the aneuploid. So basically, we were from Sam's paper on the cancer one. So we know that the micronuclei of the cells likely is going to modulate the immune system by certain pathway. So we want to kind of explore if that's also the case in the in our trophoblast cells as well. So don't get your expectations too up. We just started this, so there are not many preliminary data, but there's something. Anyway, so we hypothesized that the micronuclei in the CIN was the chromosomal instability and aneuploidy that's caused micronuclei to happen, and this is going to activate the CGAS pathway, which coordinates with STEAM and kind of modulates the immune environment. So we look at the aneuploidy, we look at the chromosome, we look at the trophoblast cells and STEAM with CGAS, and we definitely see around like 10% of the micronuclei have the CGAS protein activated. So this basically means that they can't activate this pathway. So maybe they're doing some downstream function as well, but we don't know yet. And I'm guessing like they can actually suppress the immune by this state PD-L1 pathway, the downstream, or if they're actively, constantly activated, they could be dysregulate the immune environments by prolonged activation can cause the immune to be attacked. The immune system to be more active in attacking the cells. So we believe the antibody, the right level of antibody in placenta is crucial for the immune adaptation of the pregnancies. But this is just all hypothesis right now. So the next step, we're gonna just test it in our model system. Anyway, so that says for so also we look at the transcriptome level of the trophoblasts, and we found that very interestingly, the while becoming the trophoblast cells from the pluripotent cells, there are massive downregulation in the signaling pathway involved in the cell cycle regulators. As you can see, the whole cohesion pathway is listed. So the green means downregulated, and the red means upregulated. As you can see, all those cell cycle related pathways are mostly downregulated. However, the cell proliferation is not affected. As you can see, those markers, which are red, they basically help the cells to proceed with the cell cycle. So even though they are less stringent, but the cell cycle keep going on as well. So this is probably the reason the cells are chromosomally stable and constantly producing hemoglobin. And we also look at the autophagy, since we believe that could be the regulator in the tolerance. And very interestingly, the autophagy lysosome pathway is highly activated in the placental trophoblast cells. As you can see, all those genes involved in this pathway is highly upregulated in the placenta cells. And we also do the staining and do some Western blot to confirm the level of the proteins involved in the autophagy pathways. And so we found like the lysosome degradation T genes, the system, that gene is highly activated in the trophoblast, but barely detectable in the pluripotent. So this is another confirmation autophagy definitely plays some role in this tolerance. And if we inhibit this process to go on, you can see, so basically we apply this drug, it's called QC. This drug is to block the autophagy lysosome pathway. So blocking the lysosome degradation, you can see it doesn't, no matter how high the drug level is, it doesn't affect the pluripotent stem cell proliferation. However, if the drug is high enough to totally block the lysosome pathway, the trophoblast proliferation rate is significantly decreased. So that means again the autophagy lysosome pathway is critical for the tolerance in placenta. Anyway, so from this we can conclude that the extraembryonic tissue, the extraembryonic trophoblast cells are chromosomally unstable, and anybody can be better tolerated even in those type of tissue. And apparently there's no obvious functional compromise to those cells. Anyway, so the take-home message of the day will be in early embryogenesis or early human development. It's error-prone, and they are leading to a lot of aneuploidy in those developments. And the tolerance towards aneuploidy is decreased along the way with human growth. And also aneuploidy is frequently exists in placenta and has no apparent functional compromise. And the next new hypothesis we're going to test in the future is that the broad hypothesis is if there's an evolutionary reason for this error-prone human embryogenesis, and can we harness the fitness adaptation in this process to understand more how cells compete with each other, or help the cells in certain disease type to survive, and help the normal cells to survive better than abnormal cells. So that's our bigger dream in the future. And with that, I would like to, that's all for my talk today, and I would like to thank people involved in this project and also my home institute, University of Washington, and everybody who contributes somehow to the lab establishment, new lab establishment over the past. Anyway, and with that, I thank you all for your attention, and
Questions?
yes.
Many. Many. Where do you get your samples from?
Which sample, the placenta sample?
Yeah.
So those are actually not our data. So those are the from, I think the data was from the 2014. That's a paper coming out from a Wangfeng-based lab in China. So they got a sample. So we just reuse their data. So this is a published data set which analyzed the human normal placenta, but it's not our sample, so we didn't take a sample.
So how far along the pregnancy?
So they did an 8-week pregnancy and term pregnancy. So 8-week and term.
Termination?
So for termination, not miscarriage or normal pregnancy.
You want to differentiate between chromosomal abnormalities and placenta?
So this technology of the antibody calling can actually differentiate that. So we know it's the whole chromosome change instead of just part of the genomic mutation. So we are sure from the data that those cells are the whole chromosome change.
Do you have an idea what the percentage?
So a term, as you can see, I don't know, in the cytotrophoblast cell type, you can see 20-something%. But like I said, this method is not sensitive enough to call all the accuracy, all the antibody, so it's higher than that. But also it's cell type specific as well, as you can see in the syncytial trophoblast, it's much lower. So these cells need to be very accurate, probably for their hormone production function. Yeah.
Placenta is an enormous organ.
Yes.
At term, anywhere from 250 to 750 grams.
Mhm.
How do you determine what places to biopsy?
So,
Yeah.
all right. So I don't know how they biopsy the sample, but on their data set, because they did the single cell sorting, like basically they did all the single cell from the placenta, but so we can confirm it easily the right marker for trophoblast cells. So we can cluster them, and as you can see for those, we actually by their, so this is also incorporated with RNA-seq as well. So we know these are EBT or trophoblast, the placental cells, by their transcriptome signature as well. They are expressing the right marker for the right cell type. So we know for sure those cells we are calling they are the trophoblast or extravillous trophoblast. So, but how they collect the sample, I have no clue how they do that.
This is an enormous organ.
Yeah.
Depending on where you find it.
Right, right.
And I used to give lectures to the medical students. I said, placenta is the first tumor.
Yeah.
And first tumor diagnosed. And first human cure for cancer.
Yes, self
Because
cure.
of drug.
Yeah.
For cure, because trophoblastic disease, it's so responsive to cancer.
Mm.
So, mostly in Asia, in South America, in Latin countries, their trophoblastic disease is extremely prevalent. I used to always say, it's the first cancer for humanity and the first real cure for cancer. So depending on where you find it, it can be very, very
Right. Right. I'm sure they have a systematic way to collect the right marker because for their study, they're interested in this subtype of the trophoblast. So they biopsy and then confirm with the single cell RNA they are the right trophoblast they're looking at. within their data set, there are also some other cell types like trauma cells or disabled cells on the uterus, but that can be easily screened by their technicians. Anyway, so that's a good point. So I think how they collect the sample, that definitely
This is a
matters.
big order.
Yes, yes, really, really big.
Sam, what's the percentage of solid cancer? What's the range? 60 to 80% in primary tumors.
It's your thing.
Yeah, so I have 7 questions, but I'm gonna keep them short.
You can maybe sign later. We can
No,
discuss.
actually, maybe a statement first, which I find really amazing. So, a few parallels between what you presented and cancer are really quite striking. First of all, amazing thought. The first one is differentiation. In cancer, I know differentiation is kind of not the topic, but tumors that are input and unstable are considered to be slightly more undifferentiated than the ones that are stable. So that's interesting.
Yeah.
Human cancers are way more set and input compared to mouse cancers. And that's actually a really important topic that most cancer biologists that model it in the mouse do not appreciate. That's really important. So the fact that the placenta
Evolutionary.
have different, potentially, or the mouse placenta versus human placenta have chromosome differences in their chromosomal abnormalities is super interesting.
Mm-hmm.
The third point that I'll mention is hormone-producing tumors, like estrogen-responsive breast cancer, thyroid cancer, recent 6 prostate cancer. We should even call this a cancer.
That's
They are much more stable
Oh, wow.
compared to invasive. Like the perfect example is the thyroid. The more hormone production, the more stability you get. The
Wow,
less hormone
interesting.
production, the extreme cases, anaplastic thyroid cancer. And that's much more invasive, etc. I also think it's super interesting that the invasive subtype
Yes.
of placental
Yeah.
tissue is the more unstable one because that fits very much with cancer metastasis.
Yes. Okay.
So the parallels to cancer are ridiculous, but I guess it comes back to the question of what. So, I mean, okay, we don't know if mice live for the longest humans, how much cancer they would get, right? But what is the evolutionary basis for all of this? Like, is the placenta the reason? Is the implantation the reason? Or is there something else?
I don't know, but it's somehow to do with the length of pregnancy as well as 10 months, and then I feel like it has to be like very deeply implanted to the uterus to make it supporting the long pregnancies. So I guess somehow they just adapt a way to kind of let this connection be built high enough, but not like anyway. So I think it has to do with
It's a genius model, because when you think about it, this is not just a transplant. Because if you transplant an organ, except for the liver, it remains the same size. Here you start with a microscopic thing that grows into a fetus of 7 pounds, the placenta of 2.5 pounds. The placenta with all of its abnormalities, with all of its chromosomal abnormalities, has for evolutionary reason. The evolutionary reason is, in my opinion, that we need the aneuploidy to do something for power. And
Yeah.
there is a reason why placenta and fetus grow in parallel. Because it is one thing to tolerate a little thing, it's a very different thing to tolerate a 2.5, 3.5, 7-pound baby. And whatever provides that protection needs to go in parallel. Otherwise, it couldn't handle the increase in mass. And the placenta is perfect for that. It grows in parallel. It's being thrown out, so who cares if it has aneuploidy? I mean, it seems so logical that the placenta is there not only to provide the oxygen, and not only to provide the nutrition, but also to provide the immunological power.
So you just instigated the eighth question, so that doesn't count. But the truth is, in lab mice, we have black 6 or BALC or FDD, whatever. The equivalent, there's very little genetic heterogeneity in black 6 because they're inbred strains. It sounds like Nova Scotia. And human tolerance is much broader. You have Africa, Australia, Europe, etc. So I'm wondering if mouse placenta in the wild, if you put different species of mice, or maybe the wild, etc., it would be very different because our measurements of mouse placenta is from like black 6, right, which is inbred for 10 generations. And that's where all the cancer models are most developed. But the next question I have is, in that paper, did they have any placenta previa or placenta previa that have different invasiveness into the muscle?
Which paper you on?
In that ataxia?
Oh, yes.
Did they ever have any pathological placental,
No.
placenta previa versus placenta previa?
Yeah, no. So that paper only profiled the healthy because they want to base it so much unknown about placenta. They just want to set up the standard what's normal. So they only profile the normal pregnancies. Yes.
So the epidemiological issue in that paper is that doing terminations of 8-week pregnancies, of which 25% might have miscarried occurs annually. And return placentas, which didn't express as much antibody, or survived to that point. Didn't miscarry. So it could be sort of a selection issue. It's not necessarily that the placentas correct themselves over time.
Yeah,
It might have been eliminated.
Exactly. That could be that too. So it causes a lot of bias to the sample, the data itself.
But then you have term placentas, right?
Yeah.
That is studied in nature was one.
Yeah, the somatic mutation. One more question.
Yeah, okay, nice talk. I was curious to know how stable is the aneuploidy in trophoblast stem cells?
How stable?
Yeah, if you serial passage them, the aneuploidy is always there.
We'll see. And we're gonna show you. I didn't show you this. I thought I didn't have time, but I'll just put it. So they're ultra stable. And one of the cell line you can see, they look like this, pretty much like a cancer cell line. They are like 80 to 200, 100, and they're so stable and functional just like.
Conversely, do you think the euploidy will be incompatible for those stem cells?
Usually, majority are still, for the normal cell line, majority are still normal cells. And I didn't separate them to look at them in detail, so I think the next step I'm gonna check. But I'm telling you, this cell line, they're crazy. They're proliferating so well, these cells. They're just like cancer. Look at them. They look exactly like the choriocarcinoma cell line. And they are proliferating even faster than the normal cell line. And one interesting thing we found out, those cells have harder time differentiating into the syncytial trophoblast, the hormone production one. So I didn't know about the cancer one. So thanks, Sam, for the point. But it's really amazing to me, like how they compare much parallel they can do. Just one question now. Go ahead.
Go ahead.
No, you know it's you. I was no. But go ahead, because I was
I would say in mammalian species, and based on my 20-year-old research, not as old as these small vaccines. Just want to trade that. But I wonder, it's a placenta is maternal genome. It's defective to start with. And maternal genome does not tolerate errors, because otherwise the fetus dies.
right.
And there's certain errors that are tolerated by the placenta because it's a clone. But the function of the placenta is to provide nutrition to the fetus. And the vast majority of the genes that are activated, if you look at epigenetic research, are from the paternal genome.
Yeah, the paternal trying to protect.
Exactly.
That's the evolutionary.
Maternal genome doesn't do that. It protects the fetus.
Right.
So I can see where that's the evolutionary mechanism.
Totally. Definitely needs exploring more. But anyway, so I think, thank you all, and really enjoyed the discussion.